Action of salivary amylase on starch. So first of all, take 5 ml of starch in 3 test tubes. Label the test tube as test tube number 1, 2 and 3. So I've taken 5 ml of starch in all the 3 test tubes. To this you add 3 ml of buffer. The pH is 6.7. It's the optimum pH for enzymatic activity. So in all the 3 test tubes I've taken 3 ml of buffer solution. Now in test tube number 1, you take salivary amylase. So I've taken salivary amylase in test tube number 1. Test number 2, you will take distilled water. Already starch and buffer solution is present in all the 3 test tubes. And in test tube number 3, you take dilute HCl. So test tube number 1, I have taken salivary amylase. Test tube number 2, I have taken distilled water. Test tube number 3, I have taken dilute HCl. Now keep all the 3 test tubes in water bath for around 15 minutes at 37 degree. 37 degree is the optimum temperature for enzymatic activity. So keep it in water bath for around 15 minutes. Now to each test tube you add 2 drops of iodine. So test tube number 1, I am adding 2 drops of iodine. You will not get blue color because salivary amylase has broken down starch into disaccharide. So no starch, no reaction with iodine and no blue color. In test tube number 2, you add iodine. You get blue color because starch is already present. It reacts with iodine to form blue color, that is polyiodide. So you can see the blue color. Now in test tube number 3, where dilute HCl was added, you add a drop of iodine. Here you will get umber color because HCl breaks starch into monosaccharide. So you can see the color. Test tube number 1, no blue color. Test tube number 2, you get blue color. Test tube number 3, you get umber color. So salivary amylase acts on starch, breaks starch into disaccharide, and you will not get blue color in test tube number 1. So this is all about action of salivary amylase on starch.